Dr. George Wilding. Vicepresidente y director académico adjunto de investigación clínica e interdisciplinaria del MD Anderson Cancer Center. La investigación del Dr. Wilding se centra en comprender el papel del estrés oxidativo inducido por andrógenos en la carcinogénesis de la próstata y su progresión. En su laboratorio, se desarrollaron 2 agentes dirigidos a esta vía para el cáncer de próstata, con resultados prometedores. Uno de los agentes se encuentra en pruebas clínicas y el otro se acerca a esta fase crítica. El Dr. Wilding ha publicado más de 235 artículos. Su pasión y compromiso con la educación se refleja en su experiencia por más de 20 años como investigador principal. Sin duda, una increíble trayectoria de este gran investigador. Recibamos con un gran aplauso al Dr. George Wilding.
Well, good morning, everyone. Thanks to all of you for getting up early on a Saturday morning to come and listen to us. First, I'd like to thank Luis Herrera and all of the organizers for this symposium. Once again, it's a wonderful symposium. The science for the past couple of days, in my mind, has been phenomenal and spectacular to watch the presentations. But I think the most impressive thing to me in the past couple of days have been you, the students. Your enthusiasm, your interest in science, your intelligence is very comforting to me that science for the future is going to be in good hands. And I look forward to seeing all of the accomplishments that all of you achieve. So I'm going to talk today about prostate cancer, similar to the past couple of years. And as you heard last year, we have focused for quite some time on the influence of androgen-induced oxidative stress on prostate cells, both in their role in carcinogenesis and in progression of the disease. We showed you last year that we identified at least one major source of that oxidative stress, and it was conducted through the androgen receptor binding JUMD, turning on a pathway led by spermidine acetyltransferase onto acetyl polyamine oxidase, where the reactive oxygen species are produced. Those turn on NF-kappa B, produce P52, which then combine AR, and the cycle continues in an autocrine fashion of the androgen-induced oxidative stress. We showed you that we could inhibit that, and that we screened 20,000 molecules to come up with a small molecule that would inhibit AR-JUMD or AR-P52 binding, or inhibit APAO. And if you did that, you slowed down the progression of the prostate cancer in a dish and in animals. And we continued to develop those small agents for which we hold patents and that we hope to bring into the clinic. But what I'm going to focus on today is further investigation of metabolism in prostate cancer. In the prostate cancer group at MD Anderson, there are a number of individuals, Mark Titus, Dan Frigo, others, ourselves, that are interested in metabolism. And what we've seen, and what Mark Titus has documented, is as you go from androgen-responsive prostate cancer, for which we have very good treatments, to aggressive variants of prostate cancer, for which we don't have very good treatments, and that is the state that people die from. In between, there is a transitional state where metabolism changes, and in fact, you start producing more glucocorticoid receptors. So there are significant changes that offer great opportunities for us to see targets, develop treatments, and see if we can hold the cancer in that transitional state before it gets to the aggressive variant. Today I'm going to show you some data, and it's early data. I would look at this talk as hypothesis or idea development. Contrary to last year, where you saw a series of experiments conducted over years, these are very recent data. And with the exception of posters and abstracts and talks like this, much of the data you'll see is unpublished. We wanted to look at metabolic adaptation of prostate cancer that develops when the cells become resistant to anti-androgens. So our goal in this is to establish prostate cancer, that prostate cancer cells develop resistance to anti-androgens, but in that process they shift from focused on glycolysis for their energy to oxidative phosphorylation. And that this might offer opportunities for us to try to control those resistant cells by targeting oxidative phosphorylation. So through the talk, I'm going to go through 4 different questions that we've begun to address. By no means have we completed answering each of these questions, but I'll show you some of the initial data for each of these questions. We want to know if anti-androgen resistance in prostate cancer, if the cells actually do switch from glycolysis to oxidative phosphorylation when they're treated with anti-androgens. As you all know, 100 years ago, Warburg stated that most cancers are dependent upon glycolysis for their energy. Prostate cancer cells use predominantly glycolysis. So shifting to oxidative phosphorylation may be different for them. More recent data shows that stem cells for cancer may be more OXPHOS related than glycolysis. In the second question, we'll look to see if that metabolic adaptation helps them survive when they're treated with anti-androgens. And if shifting to that OXPHOS state, whether they'll be more vulnerable if we target mitochondrial oxidative phosphorylation. And finally, I'll show you some patient-related data as to whether we can find tumors that actually seem to be expressing this shift in energy production from glycolysis to oxidative phosphorylation. So the first question is, do the anti-androgen resistant cells actually switch from glycolysis to oxidative phosphorylation? And we'll be monitoring changes in cellular glycolysis using a couple of different techniques. One is the seahorse assay that is dependent upon looking at the proton flux, which is important, especially in oxidative phosphorylation in the membrane of mitochondria, and looking at metabolites of energy production to try to quantitate it through mass spectrometry. So the first thing you see here on your left is what all of you learn in Biochemistry 101. You have glycolysis. And that's a common pathway. It produces energy. You can monitor its activity through the transition from pyruvate to lactate via lactate dehydrogenase. So it gives you a marker if you monitor lactate. On the right side of that, you see mitochondrial respiration. Much more efficient, produces many more ATP for it. But cancer cells seem to like glycolysis more. There are many aspects of oxidative phosphorylation, and you see the different modules or complexes that constitute the electron transport system. You can inhibit those different components with known drugs like rotentin, and then see what happens when you inhibit that pathway of electron transport. With the seahorse assay, there are a couple of different phases of the experiment where you serially introduce inhibitors of first oxidative phosphorylation and then glycolysis so that you shut down everything. But if you look at the left-hand side where the blue and the purple are, you can see the levels relative of how much is being produced through glycolysis versus the mitochondrial oxidative phosphorylation. And in that first part is the part that we monitor to try to determine what the cells are doing predominantly, glycolysis, oxidative phosphorylation. So when we look at this, and this will be typical readouts of seahorse assays that you'll see in papers. Sometimes they just state the OCAR, the oxygen consumption rate. But again, on the left-hand side of that, you see that with 3 different cell lines, the top line is LN caps. Those are common prostate cancer cells that are used in prostate cancer research. It was derived from a lymph node of the prostate. It is androgen responsive, produces androgen receptor, and you can see that it has a higher glycolysis rate than 2 other cell lines. The C42 cell line is actually a derivative of LN cap. So it was born from LN caps, but it is now less androgen dependent, and it is more resistant to anti-androgens such as enzalutamide. The other one, the PCA2B, is actually a patient-derived xenograft from a patient that was failing anti-androgens. Okay, so it's from a tumor that was showing resistance to anti-androgens. And you can see that both of those have lower glycolysis rates than the LN cap cells. And this just shows that if you take those data and you look at the basal-OX-phos, it's higher in the castrate-resistant prostate cancer cells, C42, or the patient xenograft compared to the LN cap cells. Okay? So it does seem that the androgen-responsive cells have more glycolysis, less OX-phos. The cells that have developed resistance tend to have more oxidative phosphorylation. So if we design an experiment and we plate the cells, wait a couple of days, treat them with the drug, in the initial experiments you'll see will be enzalutamide, which is a very common anti-androgen used clinically. Wait a day and do the experiments to look at what the state of their metabolism is. We do this and adjust for what live cells are still in the dish. Because obviously the metabolism of cells that are not live is going to be confounding to the data. So if you look at this and we take enzalutamide, and enzalutamide can inhibit the LN cap cells in single-digit micromolar levels. But if you want to inhibit 50% of the C42 cells, you have to get to 20 micromolar or higher. In this experiment, you see that the black bars are the glycolysis, and the red bars are more reflective of the oxidative phosphorylation. In this instance, when you treat with the enzalutamide, the red bars are steady, sometimes even going up a bit, but pretty clearly the black bars, the glycolysis rate is going down with treatment with enzalutamide. The glycolysis goes down with both LN cap and C42 cells. If we look at this further and do mass spec quantification of lactate and pyruvate, and very commonly you look at ratios of lactate and pyruvate to get a sense of the rate of glycolysis going on. And in this instance, we're looking at LN caps, and there is a reduced rate of glycolysis with enzalutamide. And you see that in the middle with the left-hand bars for LN cap, that when you add the enzalutamide, you see the ratio of lactate to pyruvate go down. And that is also reflected in the right-hand seahorse readout that shows a decrease in the glycolysis. So it does seem that prostate cancer cells reduce glycolysis when they are in a state of surviving treatment with enzalutamide, anti-androgens. So how does this metabolic adaptation allow them to survive better in the presence of an anti-androgen that normally would push them to not survive? So we started exploring the role of mitochondrial metabolism in self-survival under these hostile conditions. And to do that, we started using a series of stains for mitochondria. Okay? There's spectral characteristics of these dyes, and some of them reflect more of the oxidative state of the mitochondria, and some of them reflect more normal mitochondria, such as the mitotracker that you heard about a couple of days ago. In this instance, Mitosox red fluoresces upon oxidation in production of reactive oxygen species. So if you see more of this, then it means that you have higher producing mitochondria, producing in the sense of oxidative species. And if you look at examples with this, the cells, the excess oxidative species generating mitochondria, Mitosox red staining, are more in the C42 cells than in the Ellen-Cap cells when you treat them with enzalutamide. Now you see the difference in doses here because we tried to give equivalent inhibitory doses, and we used 10 for the Ellen-Cap cells, more sensitive, than the enzalutamide cells where we used 20 micromolar. But you can clearly see here that the mitochondria of the C42 cells, that there is much more red intensity of the staining. So there are many more mitochondria that are producing reactive oxygen species. Now that can do a number of things to mitochondria. If you're producing all of these reactive species, you're going to create damage. And cells don't like to have damaged mitochondria. So they have a way of processing those. That if the mitochondria develops enough damage, it undergoes a process called mitophagy. And how do we try to measure mitophagy? Well, it's in this instance, in the other day, you heard the FRET assay being used in a different circumstance. And this is just a cartoon explaining the FRET assay, where you have a first protein. In this instance, you're going to see that we're using a protein that is part of the lysosomal membrane complex. And you attach an antibody to that is excited at a certain wavelength, and then it emits at a different wavelength. Then you take your second protein. In this instance, it would be the Mitosox red label of the mitochondria, and you label that with an antibody that is excited at the emission wavelength of the first. And then it emits a final wavelength that you can measure. So only if those two proteins are very close together do you get that sequence of excite at one wavelength, emit at another wavelength, which turns on the emission of a third wavelength that you can measure. And when we do that with the various cell lines, and you can see the various treatments, control, enzalutamide, R1881 is a synthetic testosterone, or the combination of those. You can see that with Ellen-Cap cells, there's a little bit of difference, little bit of increase in the red bar. But in the two cell lines, and in this instance, with the CWR, that is another enzalutamide-resistant cell line, and the C42 cells, that clearly the red bar is increased. And that means that the mitochondria that are producing all the oxidative species and the mitophagy lysosomal complex are very close together, and that you have an increase in mitophagy. And this is just a cartoon showing that process where the lysosomal complex comes together, the membrane envelops the mitochondria, and eventually you have mitophagy occurring to rid the cell of that mitochondria that is not functioning very well. Well, you can see with the LC3B, that's the protein that we use in Fred. Well, what happens if we silence that and shut off the mitophagy pathway? And you see in this instance, with the diagram on the left, the bar graphs on the left, that in the middle, that if we silence LC3B, we actually make the cells more sensitive to the enzalutamide, as you would predict. And you see that difference in the right as to how it is characterized. So we can show that if we inhibit the mitophagy, that the cells that are resistant to enzalutamide become a bit more sensitive to the enzalutamide. Well, using a different dye, in this instance, the MitoTracker Orange, that you saw some examples of the other day, that tracks normal mitochondria that are functioning well and are not damaged. So you can stain and you can see how many mitochondria do we have, where are they, what's their shape, etc. So when you look and you stain cells with that, and you look at Ellen Cap treated with enzalutamide versus the C42 cells, in the Ellen Cap cells, you see many mitochondria that are very typical of what you see in your textbooks. The bean-shaped mitochondria that are similar in shape to each other, relatively small. But what you see in the enzalutamide-treated C42 cells is denoted by the arrows. You start seeing these very long mitochondria. And that is a process called fusion. Mitochondria can undergo fission, where they kind of break up into smaller mitochondria, or fusion, where they join together and form these long mitochondria. And you've seen some examples of that presented to you yesterday and the day before. So what is happening with this? In that we have an example, on one hand, that when you treat with anti-androgen in cells that are resistant to the anti-androgen, that they start chewing up and getting rid of damaged mitochondria, mitophagy. But on the other hand, we have pictures of mitochondria joining together and forming these elongated mitochondria that appear to be more functional. And how do these two things go together? They seem to be variant to each other. So if we do the Fred analysis, but this time we use MitoTracker Orange instead of Mito Red, so we're staining normal functioning mitochondria instead of the ones with high oxidative stress. What happens with the rate, or what do we see with mitophagy? And in fact, you see with the red bars, with the Ellen Cap cells, it may go up a little bit, but not significantly. And with these normal stained mitochondria in the C42 cells, when you treat with enzalutamide, the red bar doesn't go up, it actually goes down. So actually the rate of mitophagy in that set of mitochondria goes down, contrary to what happens to the damaged mitochondria. In fact, there's some publications just recently in the JBC by another group that looked at mitochondrial localization, import, and function of the androgen receptor. So what is happening there? We think of androgen receptor, AR, ER, GR, etc., as being nuclear hormone receptors, go to the nucleus, turn on a bunch of genes. What is the AR doing in the mitochondria? That's completely contrary to some of the things that we normally think of AR. So in fact, what we did since we had been working on the AR function, and we decided, well, let's do a FRET analysis of looking at mitochondria. In this instance, we'll use the mitotracker orange and target the normal functioning mitochondria. But instead of using for the first target the lysosomal complex, we'll use androgen receptor as a target and get an antibody to that then will emit, when excited, at a wavelength that will turn on the antibody tied to mitotracker orange. And you see here in the upper left that the purple stain, blue stain, is a DAPI stain. And when we do the excitation of the labeled AR, you don't see any FRET occurring, okay? Because you only have that one. When you do mito alone, you don't see any FRET. So if you look at mito orange alone, no FRET. AR alone, no FRET. You put them together, and on the bottom, and you do the excitation in the two antibodies present, you see the FRET excitation is shown in the bottom right panel. So somehow the AR is getting involved with this. And this is very fresh data that we have, and we're beginning to pursue this pathway since there are publications in the past that the AR is involved with the mitochondria. So how can we try to put this together? Since we seem to be seeing two different things going in different directions. We see mitochondria fusing, and we see mitochondria being chewed up through mitophagy. The key here is the oxidative phosphorylation, that if you increase OXPHOS, you will increase ROS. And there are two ways that the cells, from a mitochondrial perspective, can deal with that. That if the ROS damages the mitochondria too much, you undergo mitophagy and get rid of it. If you damage it a little bit, then the other way that cells can deal with mitochondrial changes is by fusing. You take an undamaged mitochondria and one that is functioning but has some damage, you fuse them and you dilute the damaged parts, and you have a better functioning overall mitochondria, but it also looks longer because you've had the fusion. And we seem to have both of those things going on in the cells that are treated with anti-androgens. They deal with it by getting rid of damaged mitochondria and diluting out others through fusion. So we've worked with two of our colleagues at MD Anderson, Dan Frigo and Cullen Taniguchi. And we've gotten from Cullen some of the vectors for expressing or blocking proteins that cause fusion or fission. And we're in the midst of experiments, since now we have these vectors, that what will happen to the cells if we inhibit fusion from occurring? Will they become more sensitive to enzalutamide anti-androgen treatment? And that's a series of experiments where this set is ongoing. There have also been other publications, and this is from a colleague up at the University of UT Tyler, and they've shown that when you inhibit or change complex 1, it results in enhanced mitochondrial fusion, and that's associated with prostate cancer progression. So that's another group looking that is seen that if you have fusion of the mitochondria, that's associated with prostate cancer cells that are showing more progression, just as you might expect prostate cancer cells to do if they're resistant to anti-androgen therapy. So we're beginning to see that metabolic switch provides a survival advantage by altering the mitochondrial ROS, creating fusion, changing mitophagy, and that the AR might be involved in all of this. So will the cells in this state, can we take advantage of that? Are there targets that we can use that if the cells that are becoming resistant, can we target something to kill those cells or make them more sensitive to the enzalutamide? So here's a common picture of the electron transport with the various complexes. And you can see that there are a number of drugs that have been shown to inhibit complex 1, 2, 3, etc. And MD Anderson, in our drug development group, you see IX 10759. That's been shown to inhibit complex 1. And there are a number of publications with that. We're in the midst of a phase 1 study of this drug. It's actually towards the end of the phase 1. We're approaching, we think, a phase 2 dose. There have been some prostate cancer patients on the phase 1 with some interesting results, and we're hoping to have an expanded phase of this drug in prostate cancer. Here's some of the other drugs, and there are many more. We could make a very long list of drugs that are known to inhibit various parts of the complex. So if we take, again, the LNCAP cells, C42 cells, enzalutamide sensitive, enzalutamide resistant cells, and we treat them with the IX compound that inhibits oxidative phosphorylation. As you expect, you see the red bars that are reflective of OXPHOS, and they go down with increasing concentrations of IX. Cells normally compensate that if you inhibit OXPHOS, they do more glycolysis. They need the energy. So you see one goes down, one goes up. And you see that in both instances here. You can see that we went up to 10 nanomolar. And that's actually a clinically achievable concentration that we're seeing in the blood of patients in the phase 1 study. So low concentrations, nanomolar, compared to micromolar of many drugs that you all take, but achievable clinically. So doing experiments, plate the cells, wait a couple of days, treat with the drug, and if we're doing 2 drugs, a day later add the second drug, and a day later look at the metabolism. Again, normalized to living cells. And in this instance, we look at the combination of enzalutamide and IX. So we're giving enzalutamide and we're giving IX. In what direction does everything go? Well, you see that the glycolysis in the C42 cells remains high and the OXPHOS goes down. Same thing when you treat with IX alone. That's the pattern that you expect. On the right panel, when you treat with IX plus enzalutamide, Glycolysis can't go as high as you expect, and you still decrease oxidative phosphorylation. So you hold down the glycolysis and you push down the OX-phos. Let me just show this then. So what happens when you combine these and you actually look at growth? Well, in the L-cap cells, adding IX to enzalutamide, the cells are already sensitive to enzalutamide, and the IX didn't really add very much. But if you take the cells, the C42 cells on the right, and you add IX to the enzalutamide, you see a significant improvement in the reduction of the growth of the cells. So the combination is more potent than enzalutamide alone. And that's what we were hoping for, is that given all the metabolic changes, if we target the right thing, target OX-phos, that we actually could enhance the effects of enzalutamide. So it does seem that cells treated with enzalutamide might be more vulnerable to mitochondrial metabolism inhibitors. Can we actually find tumors that reflect this other than cell lines? So we started exploring mitochondrial metabolism in circulating tumor cells from patients that are treated with a variety of treatments that might be responding to anti-androgens or failing anti-androgens, and on patient-derived xenografts that come from patients that might be prior to treatment, during effective treatment, after treatment stopped working. Nora Navone at MD Anderson has been collecting these PDXs. She has over 100 now that she has characterized, genomic profiling, et cetera, growing them in mice. Some of them have turned into cell lines. And so we started looking at the PDXs as well as CTCs. As far as the CTCs are concerned, you've seen talks on isolating CTCs yesterday. We work with Dr. Rubin in Amado Zarita. Amado was here last year. He presented to you much of his data that he, working with Nick Navin, a molecular biologist at MD Anderson, they've done extensive work on CTCs and also small clusters of cells in the bone marrow of patients with various states of prostate cancer. He's done genomic sequencing and profiling in those CTCs. That profiling includes a number of genes and enzymes that are related to mitochondrial metabolism. So all of that has been done, and we're now, given what I've just shown you, going back and reanalyzing the data to look at mitochondrial metabolism changes in all the work that Amado has done. So if we take CTCs from patients treated with enzalutamide, and we look at mitosox fluorescence, in this instance, the mitosox red, so we're looking for cells that have mitochondria that are producing a lot of oxidative stress, a lot of ROS. And we do that, we stain with the mitosox red, and then we segment it and try to judge what the level of intensity is. And on the right, you see a panel of patients that we've started to collect. And in the first 15 or 16 patients, those are all patients that are responding to enzalutamide. And you see that they have a certain level of the mitosox red staining and intensity. If you look at the 4 or 5 patients at the other end, those are patients that are failing enzalutamide. And just as we saw in tissue culture, we're seeing an increase in the number of mitochondria that are staining for mitosox red, high ROS. What are the 2 red circles there? Well, in the first red circle, that's not two different patients. It's actually the same patient who still is responding to enzalutamide, but you can see the second bar is up a little bit from the first bar. This patient is still alive and is actually due to come back in clinic this month, and we'll see if he continues to respond to enzalutamide and what his CTCs are showing. In the second circle, you see that the first bar is down at the level of enzalutamide response. And in fact, that patient was responding to enzalutamide at the time. The second bar in that circle is significantly higher, and that's when clinically he was failing enzalutamide. So again, these patient CTCs are corresponding with what we're seeing in tissue culture. Unfortunately, this patient succumbed to his disease, so we won't be able to get subsequent samples. Oop, what happened there? Okay, so what happens if we take those CTCs? Can we actually try to do the FRET analysis on them? And remember, when we did the FRET with LC3B associated with mitophagy, and performed that on the C42 cells, we saw increased mitophagy. So here's a snapshot of some of the patients that were on the previous bar graph. You see the two samples where patients were responding to enzalutamide, and the FRET analysis shows low activity. And then in the patients that are failing enzalutamide, the FRET activity goes up. The mitophagy goes up. And again, you see the red circle. That's that patient where on the left he was responding, and on the right he was failing enzalutamide. So in patient CTCs, we can reproduce seeing this increase in mitophagy as cells become resistant to the treatment. We also wondered whether we can actually get CTCs from PDX models. Put the PDX in the mouse, try to see if you can pick up human circulating prostate cells. I have to admit that when my lab raised this to me, I had my doubts. It's like, seriously, you're going to be able to pick these up in a mouse of a PDX implanted in the mouse. But in fact, we are picking up human prostate cancer cells circulating in the blood of mice that we're putting the PDXs. And this obviously is another path of experimentation that we're going down, that we have an animal model using human xenografts that have shown to be sensitive and resistant to enzalutamide, and gives us another model to do co-clinical trial type work. So we do see that mitochondrial ROS and mitophagy analysis can be done in clinical samples in PDX models. And it might provide us with the model of identifying metabolic changes, mitochondrial metabolic changes in patients as well as animal models. So how might we eventually try to clinically apply this? As you see, much of these data are early. As I said at the beginning, I would view this as really idea generation, hypothesis generation, rather than we have enough data to form the hypothesis, let's keep going. So it's early in our exploration of this. But we have been able to standardize CTC assays to look at metabolism and how it's switching. We have been able to identify patients who might best respond to mitochondrial targeting in the treatment, and eventually, with many more samples that we can get from our clinical colleagues, hopefully we can look and see whether this is a PD marker of patients responding to treatment who are about to fail treatment and whether we can do something to preempt that. So with that, I'll acknowledge the number of people that I've commented on through the talk. Of course, we're grateful to all of our patients and our colleagues in the various forms of funding that we've received for this experimentation. Thank you.